Mendelian inheritance patterns and laws of heredity. Early in the history of mankind, man could see and observe clear characters in their offsprings which closely resemble their own. However, how these traits are passed on from parents to offsprings was not understood. Many theories were put forth based on the assumptions and religious beliefs to try to explain how characters are transmitted from one generation to another. But none of these had any practical proof or convincing reasoning. It was then in the 16th century when the Austrian monk Gregor Mendel carried out systematic observations on generations of pea plants that he was growing in his kitchen gardens. He applied his knowledge of statistics to the observations he had made on the pea plant characters, and thus he came to definite conclusions regarding inheritance. He analyzed the pea plant characters generation after generation. Gregor Mendel could scientifically explain how parents transfer hereditary characters to the young. He put forward simple laws and basic concepts to explain inheritance. From the year 1904 and 1906, Gregor Mendel's experiments and the laws based on them were rediscovered and given scientific recognition. Hugo de Ries, Erich von Schmock and Karl Correns verified the basic concepts, republished his research, and hence till today these theories are widely expected as the foundations of understanding the science of genetics, the study of inheritance. Gregor Mendel is consequently known as father of genetics. All leading discoveries and developments in the field of genetics has been achieved because of these foundations and principles given by Gregor Mendel. Let us carefully follow his main experiments and understand the laws which he gave us based on this pioneering research. Gregor Mendel selected the pea plant Pisum sativum for several reasons. He chose the garden pea because it was self-fertilizing, having both male and female gametes. These develop on different parts of the same flower where self-fertilization takes place. This made it easier for Mendel to see a character in the parent and then in the next generation. These plants therefore breed true for certain traits in many generations. There is no involvement of outside or new characters after fertilization. Therefore, successive generations are just like their parents in one or more traits when the offsprings are grown from seeds of self-fertilized plants. Mendel was also aware that Pisum sativum plants could also cross-fertilize when the gametes from different plants are allowed to mix under controlled conditions. Mendel could therefore track clearly observable traits through many generations. He observed definite patterns regarding the inheritance of such traits and the control of the hereditary material passed on to the gametes. Mendel focused on studying the hereditary characters one at a time as they were passed on from generation to generation. He selected clearly discernible traits of Pisum sativum where the change or difference could be apparent of after transmission. The traits he observed were color of the flower, that is purple or pink, shape of the seed, round or wrinkled, color of seed, yellow or green, pod shaped, inflated or depressed, pod color, green or yellow, flower position, axillary or apical, stem length, tall or rough. Mendel performed his experiments systematically. Tall plants and rough plants were fertilized and then allowed the next generation to self-fertilize and observe the trait for successive generations. This was later called as his monohybrid experiment as he selected only one trait. In the next step, he took two distinct traits in separate plants and allowed them to cross-fertilize. In subsequent generations, the hybrids were self-crossed. They followed both traits simultaneously through successive generations. We now know these experiments as his dihybrid cross. Mendel was a mathematical genius. He carefully counted the number of plants involved in each breeding experiments. He also systematically recorded the numbers of each character observed in each generation and applied statistics to the outcome of each experiment, and hence he was able to draw valid conclusions. Mendel's monohybrid experiment. For the monohybrid experiment, Mendel selected plants with one distinct character. He performed a cross between varieties exhibiting alternative forms of this trait. For example, he removed the male part of the flower of a plant that produced white flowers and fertilized it with the pollen from a plant having purple flowers. These were the parental generation. Then he took single characters for the spe generation of pure variety, which were known to breed true for several generations, giving the same traits. Mendel then observed that the hybrid at the first, that is F1 generation, produced all purple colored varieties, suggesting that purple was the dominant expression of this trait. Finally, he allowed the F1 hybrids to self-fertilize, and in the second filial generation F2, he observed that there were 3 times as many of the dominant character as there were of the recessive character. He counted the number of offsprings and obtained plants with purple colored flowers in a ratio of 3:1 over plants with white colored flowers. He performed similar monohybrid experiments, selected different contrasting traits, one trait at a time. For example, at the F2 generation, he obtained tall and dwarf plants in a 3:1 ratio. Mendel allowed F1 generation plants to self-fertilize and found that 1/3 tall plants of the F2 produced only tall plants, 2/3 tall plants of F2 produced both tall and dwarf plants in a 3:1 ratio at F3. Therefore, putting symbols for each unit and factor, that is now termed alleles or genes, he obtained results as depicted. Based on these experiments with each pair of contrasting characters, Mendel was able to observe that only one of them appeared in consistently larger numbers at F2, and at F1 it is a character which is expressed while the other contrasting trait remains masked. Thus, the character observed at F1 is called the dominant character. The character which remains unexpressed at F1 is called the recessive character. However, every trait was represented by its factor which appeared and could be seen in the second generation. A character may remain masked but is surely not lost in any generation. It can be expressed and seen in the ensuing generations. After many such cross experiments were carried for a single character at a time, Mendel then considered 2 pairs of contrasting characters at a time. This was the dihybrid cross. Dihybrid cross. After Mendel's success with the monohybrid cross, he took 2 pairs of contrasting characters at a time in the same plant, the garden pea, and studied their pattern of inheritance in relation to each other. He chose 2 traits together: plant height, flower color, seed color, seed shape. Mendel therefore carried out experiments focusing on 2 phenotypic traits, that is 2 genes at the same time. He selected garden pea plants with round shaped yellow colored seeds which bred true and crossed them with plants having wrinkled green colored seeds. At the F1 generation, he found plants with round shaped yellow colored seeds. These results were the same as the expected based on his monohybrid crosses. Therefore, he knew that each round trait was dominant over wrinkled and yellow seeds were dominant over the green variety. Hence, the yellow round varieties here too showed dominance. Taking Y for yellow and small y for green, as well as capital R for round and small r for wrinkled, the genotype of the parental, that is capital P generation, where capital R, capital Y cross with small r, small y, small y. Thus, the self-hybridized F1 plants at the F2 generation yielded 3/4 of the plants with yellow traits, round shape, and 1/4 of the having green or wrinkled traits. In the dihybrid cross, the phenotypes appear in a ratio of 9:3:3:1. 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Mendel thus concluded that each character appeared in the second generation and that no character was lost or mixed with any other. The proportion in which these characters appeared reflected on their dominance within the population. Mendel's laws. Based on these observations from his experiments, Mendel also put forth two important laws. These laws are widely accepted even today as the laws of inheritance, since Mendel's experiments and his laws have defined inheritance and helped us to understand how characters are passed from generation to generation. First law of inheritance, the law of segregation. This law states that in the cell, the two units of a factor which control a trait remain together throughout life, from zygote to gametogenesis. During gametogenesis, that is formation of sex cells, these units separate or segregate from each without mixing, so that each sex cell has only one unit, either dominant or recessive for the given trait. Thus, the female produces gametes with half a set of units and the male produces gametes with 1/2 of a complete set of units or factors. During fertilization, both units come together through the gametes so that the next generation receives a complement of units from each parent. Later, a geneticist, Johansen, coined the term genes for the word factors used by Mendel, and the term allele was later coined for the units that make up a factor controlling a trait. Second law of inheritance, the law of independent assortment. Based on the results obtained from dihybrid cross, Mendel made an important observation and gave the law of independent assortment. The law states that when two independent traits controlled by two separate distinct genes or factors are inherited together during the formation of gametes, the genes for each trait are segregated independently. This means that each unit, that is allele, of the gene is distinct and is passed on to the gametes without any interference from the other. One gene pair is inherited by the next generation independent of the other, and the units of each gene has equal chance of entering the gamete and being inherited and expressed in the next generation. Accordingly, therefore, when the gametes are formed, the alleles, that is the units of different genes, that is factors of the parents, separate or segregate independent of each other. members of each gene pair to not influence each other but sort out independently, so that all possible combinations of gene satisfactors can be found in the progeny. Thus, as the father of genetics, Gregor Mendel systematically understood the inheritance of traits and formulated two basic laws to understand this phenomenon. From these very beginning grew one of the most evolved and investigated sciences of the 21st century, genetics. Mendel's law can thus be applied for understanding the inheritance patterns for characters, which have distinct dominant and recessive traits, and the other which are determined by a single gene. The genes are on separate chromosomes, the genes are on separate homologues of the same chromosome. The database online Mendelian Inheritance in Man provides information on 730 known disorders which follow the Mendelian pattern of inheritance. Patterns of inheritance. Mendel's studies of inheritance patterns in pea plants are a solid foundation for our current understanding of single gene characteristics, whether they are inherited in a dominant or recessive manner, or whether the gene lies on an autosome or a sex chromosome. Accordingly, Mendel identified 4 distinct patterns of inheritance. Traits are inherited in a manner that may be autosomal recessive, autosomal dominant, sex-linked recessive, or sex-linked dominant inheritance patterns. Autosomal recessive inheritance. For a certain trait, when an individual inherits a recessive allele from each of his parents, this genotype has 2 recessive alleles and it is known as a homozygous recessive condition. The person does not show the dominant trait and will instead express the masked recessive character. The inheritance of such traits is very simple. If both parents carry the gene in the heterozygous condition, that is capital A and small a, then any offspring of theirs has a 50% chance of having a heterozygous genotype inheriting one copy of the recessive allele. Their offspring also have 25% chance of inheriting both recessive alleles, homozygous recessive form, and thus manifesting the recessive trait. Moreover, if both parents have a homozygous recessive genotype, any offspring of theirs will be homozygous recessive, showing the recessive character. When these recessive alleles are located on the autosomes and inherited such that the recessive trait is seen in the offspring, it is termed as an autosomal recessive inheritance. There are certain disorders caused by a change in a single gene. When such a recessive allele is located on the autosome, it follows an autosomal recessive pattern of inheritance. These single gene autosomal recessive disorders occur only in individuals with 2 mutant alleles of the disease associated gene. Remember, for any given gene, a person inherits one allele from his or her mother and one allele from his or her father. Therefore, individuals with an autosomal recessive single gene disease inherit one mutant allele of the disease associated gene from each of their parents, and hence both parents are carrier. Pedigrees of families are drawn up using specific symbols based on the history of the disorder in the generation to study the trend of inheritance of the disorder in families. Such pedigrees reveal that with multiple affected generations, autosomal recessive single gene disease often show a clear pattern in which the disease skips one or more generations. If one parent has the disorder with two recessive alleles and the other parent is normal, the offsprings may only inherit one copy of the defective allele, and hence that generation does not show the disorder. Phenylketonuria, that is PKU, is a prominent example of a single gene disorder with an autosomal recessive inheritance pattern. PKU is associated with mutations in the gene that encodes the enzyme phenylaniline hydroxylase PAH. When a person has this mutation, PAH enzyme is non-functional. So, he or she is unable to convert the amino acid phenylalanine to tyrosine, which is an essential building block of dietary proteins. As a result, individuals with PKU accumulate high levels of phenylalanine in the urine and blood, and this build-up eventually causes mental retardation and behavioral abnormalities. Several other human diseases including cystic fibrosis, sickle cell anemia and oculocutaneous albinisms also exhibit an autosomal recessive inheritance pattern. Cystic fibrosis is associated with recessive mutations in the CFTR gene, whereas sickle cell anemia is associated with recessive mutations in the beta hemoglobin gene, that is HBB gene. Autosomal dominant inheritance. A person carrying even only one allele for the dominant trait, heterozygous condition, will express the features of a dominant character. If the dominant allele, if passed on to the offspring, it will show the dominant trait, even if the allele received from the other parent is recessive. A dominant trait typically appears in each generation since the allele is expressed even by heterozygotes, capital A and small a. If one parent is heterozygous, that is capital A small a, and the other is homozygous, recessive, small a, there is a 50% chance that any child of theirs will be heterozygous, capital A small a. Since such traits are expressed both in the heterozygous and homozygous condition, they are commonly seen in the population. Autosomal dominant single gene disorders occur in individuals who have a single mutant copy of the disease associated gene. The gene locus will be one of the autosomes. In this case, the presence of a single non-mutant or wild type copy of the gene is not enough to prevent the expression of this disorder. Individuals can inherit the mutant copy of the disease associated gene from either an affected mother or an affected father. All four modes of transmission are seen. Huntington's disease is a progressive neurodegenerative disorder characterized by progressive deterioration of nervous coordination and increased involuntary movements, Huntington's chorea. It is a well-known example of an autosomal dominant single gene disease. Most individuals with a single copy of the mutant Huntington disease, HTT, will have Huntington's disease later in life. In the case of Huntington's disease, however, the late onset of the disorder means that many affected individuals have already had children before they are affected by the dominant allele. Myotonic dystrophy, familial hypercholesterolemia, polycystic kidney disease serve as additional examples of autosomal dominant single gene disorders. Sex-linked inheritance. The inheritance of a trait, that is phenotype, that is determined by a gene located on one of the sex chromosomes. Genetic studies have shown that such traits exhibit characteristic patterns of familial transmission. Sex-linked traits are distinguishable by their mode of transmission through successive generations of a family. In humans, it is preferable to speak of such inheritance in terms of X-linked or Y-linked inheritance. X-linked recessive inheritance. Genes on the X chromosome control many traits related to vision and blood biochemistry. Such traits manifest themselves differently in males and females. Since males have only one copy of the active X chromosome, males therefore show a recessive phenotype more frequently than the females. Since females may inherit a dominant or normal gene from the X chromosome of the mother, a son cannot inherit a recessive allele from the father, but a daughter can. Hence, father to son transmission is not observed. A daughter who inherits a recessive, that is defective allele from the father is termed a carrier, since she carries this allele to the next generation such that each of her male offsprings have a 50% chance of inheriting the recessive allele. Red-green color blindness was the first human trait proven to be due to a gene on the X chromosome. Males are more frequently affected than females, having only one X chromosome which may have the recessive allele. Females who have one copy of the mutant gene, that is, are heterozygous or carriers, are not at all affected since they have a dominant allele on the second X to express the normal trait. Among offsprings of carrier mothers, their sons have a 50% chance of being affected, whether their daughters have a 50% chance of being carriers. Affected fathers cannot pass their X chromosome linked recessive allele to their sons, but do pass it all to all their daughters who thereby are carriers. The two forms of hemophilia, Duchenne muscular dystrophy and glucose 6-phosphate dehydrogenase deficiency, that predisposes to hemolytic anemia are some of the commonly known disorders inherited through this mechanism. X chromosome linked dominant inheritance. There are fewer dominantly inherited forms of human disease which are X chromosome linked. Females with an X chromosome linked dominant disease can inherit the mutant gene from either an affected mother or an affected father, whereas males always inherit such diseases from an affected mother. Examples of X chromosome linked dominant diseases are rare. For instance, dominant mutations in the phosphate regulate endopeptidase gene PHEX, which has its locus on the X chromosome and is associated with the X-linked dominant hypophosphatemic retics. Similarly, Rett syndrome, a neurodevelopmental disease, is associated with dominant mutation in the methyl CpG binding protein 2 gene MECP2. Rett syndrome almost exclusively affects females because male embryos with a dominant mutation in the MECP2 gene rarely survive. Y chromosome linked inheritance. Y chromosome linked traits are governed by the father to son transmission by the allelic, since the gene lies on the Y chromosome. This excludes any possibility of a female inheriting such traits or disorders except in rare translocation anomalies. It is well known that only males have a Y chromosome and they receive their Y chromosome from their father. Y-linked gene, single gene diseases are always passed on from affected fathers to their sons. It makes no difference whether the Y chromosome linked mutation is dominant or recessive, because only one copy of the mutated genes is ever present. Thus, the disease associated phenotype always expresses itself. One example of a Y-linked disorder is non-obstructive spermatogenic failure. a condition that leads to infertility in males. This disorder is associated with mutations in the ubiquitin specific protease 9Y gene USP9Y on the Y chromosome. Hairy ear rims and hairlines forms are traits often associated with the Y chromosome.
Thus, hereby we conclude the session that has been talked so far. The true concept of how characteristics are passed on from parents to offsprings was unknown, and a large number of false theories were being propounded to explain inheritance. It was in the 16th century that the Austrian monk Gregor Mendel carried out systematic experiments on generations of pea plants, applied his knowledge of statistics to his observations, and thus came to definite conclusions regarding inheritance. Gregor Mendel carried out a monohybrid experiment, taking one trait at a time, based on which he put forward simple concepts of factors, that is genes, having two units, that is alleles, which show dominant and recessive interactions to explain inheritance. He then focused on two characters in his dihybrid experiment. Based on these observations, Mendel put forth the laws of inheritance. Mendel's experiments and his laws have defined inheritance and helped us to understand the patterns through which characters are passed from generation to generation. Gregor Mendel is consequently known as father of genetics. Mendel's studies of inheritance patterns are solid foundation for our current understanding of single gene characteristics, whether they are inherited in a dominant or recessive manner, or whether the gene lies on an autosome or a sex chromosome. Accordingly, Mendel identified four distinct patterns of inheritance, that are autosomal recessive, autosomal dominant, sex linked recessive, or sex linked dominant inheritance patterns. The database Online Mendelian Inheritance in Man provides information on 730 known disorders which follow the Mendelian patterns of inheritance.